Good morning, children. Good morning, ma'am. In yesterday's class, we had completed about the different types of blood groups. Today, we are going to demonstrate one activity, identifying your own blood group in our classroom. Okay, how can you identify your own blood group in the classroom? With the help of some kit is there by using Antisera, and along with some materials are needed for doing this activity. The materials required to perform this blood group activity are porcelain plate, which should be very neat. You have to wash it and you have to dry it with the cloth, and after that you have to draw 3 circles on the porcelain plate. This is circle A, this is circle B, and this is circle RH. Means in this circle we are using Antisera A, in the second circle we are using Antisera B, and in the third circle we are using Antisera RHD. So that is the reason I have divided the porcelain plate into 3 sections. In that I am drawing 3 circles, one is representing A, one is representing B, and another one is representing RHD. And 3 reagents are required. One is Antisera A, Antisera A, the second one is Antisera B, and the third one is RHD, Antisera Anti-D. So these are the Antisera bottles which are used to identify your blood group, and whereas this Antisera RHD, it is used to identify your blood group either it is a positive or negative, because before you are performing this activity, this spirit is must, and even you need one cotton balls, okay, and 3 sticks which are used to mix the blood with Antisera, as well as needles, syringe needles which are used to prick your finger, and each needle should be used separately for one children. It should not be used for next person. After using it, you have to dispose in the basket. Never use the same needle for 2 or 3 persons. Okay. With the help of wax pencil, divide the porcelain plate into 3 sections. One is, and draw 3 circles in it. One is representing Antisera A, and other circle is representing Antisera B, and other circle is representing Antisera RHD. So use a wax pencil, because to understand the reaction very clearly, we are using in a different circles here. Okay. Next thing is that now take the Antisera A bottle. Place 1 drop of Antisera A on the porcelain plate. 1 drop of Antisera A within the circle of A. Next, take Antisera B bottle. Place 1 drop of Antisera B in the second circle which is representing B. Next, take Antisera RHD, Anti-D, and place 1 drop of RHD in the third circle which is representing RH. Now choose a finger from your left hand, especially the ring finger, children. Okay, and take the surgical spirit. Before doing this activity, first we have to clean the finger with a spirit. So dip this cotton ball in the spirit and clean the area where we are pricking on it with the help of needle. So clean with the surgical spirit. Then take the surgical needle. You have to use only this needle only for one time, children. Okay. After that, you have to throw it in dustbin. Wrap in the paper and throw it in dustbin. Then prick on the finger with the help of needle. Gently press the finger. The blood is oozing out. And place this finger on each circle. In circle A, in circle B, in circle RH. Here we have to use 3 separate sticks for mixing Antisera and the blood. Okay, you should not use, this is the precautionary measure, you should not use the same stick for same blood group. Okay, so for each circle you need 1 stick. Now you mix Antisera A with the blood sample. Now carefully keep aside and take another stick and mix Antisera B with the blood sample. Mix it thoroughly and take one more stick and mix Antisera RHD with the blood sample. And wait for few minutes. Now after few minutes, after completing or 5 minutes, you can clearly see in the blood sample some clumps are formed. Okay. You have to check each and every sample whether agglutination occurs or not. In the A sample, you can see clearly the agglutination, the clumps are there. Okay, and in the B sample also you can find. Have you understood? If it does not show agglutination with neither A nor B, that blood group is O. If it is showing agglutination with A as well as B, you call it that blood group as an AB blood group. And this RH is used to identify whether your blood group is positive or negative. So here you can see the grainy clumps in the sample, that will indicate that it is a positive blood group. Have you understood all of you? Yes? So in this way you can identify your own blood group with the help of these Antiseras. Antisera A, Antisera B and Antisera D. Yes, did you understand? So these Antiseras are the main reagents to conduct this activity. And this activity is also very important thing because you can do your own, you can identify your own blood group with the help of the reagents. But care should be taken when you are using the needles. Okay, whenever you are using these needles, okay, surgical needles you have to use, and one thing is that use only one needle. And don't use this needle for each and every person, children. So you have to use it and you have to wrap in the paper and you have to throw it in a dustbin. Okay. Have you understood all of you this activity?